Welcome to Fundamentals of Genetics. Today, we are diving into the fascinating world of physical and chemical mutagens. We will explore how certain environmental factors can alter the very blueprint of life, our DNA. By the end of this session, you will understand exactly how these agents work and how we measure their impact. Let us get started. To begin, imagine you are copying a very long, complex recipe book by hand. If you are just tired, you might make a random spelling mistake once in a while. That is like a spontaneous mutation. But now imagine someone bumping your elbow or spilling coffee on the pages. These external disruptions force you to make many more mistakes. In genetics, the recipe book is your DNA, and those disruptive external factors are called mutagens. They are environmental agents that physically or chemically damage the DNA, causing the cellular machinery to make errors when trying to read or repair it. Following up on that definition, it is important to distinguish between the types of mutations. Spontaneous mutations occur naturally due to normal cellular processes, like occasional DNA replication errors. However, induced mutations are entirely different. They are caused directly by these external mutagens. Mutagens interact directly with the DNA molecule itself. They can alter its structure by breaking its sugar-phosphate backbone, changing the chemical properties of its bases, or even inserting themselves physically between the bases. This direct interference is what forces the cellular machinery to make so many unnatural errors. Now that we understand how they interact with DNA, why do we care about them? In the real world, understanding mutagens is the absolute foundation of cancer biology, toxicology, and environmental safety. Because mutations are changes in the genetic code, mutagens are often also carcinogens, meaning they can cause cancer by disrupting the genes that control cell division. By identifying these hazardous substances, we can regulate them in our food and workplaces to protect public health. On the flip side, geneticists actually use mutagens intentionally in the laboratory. By deliberately breaking specific genes, scientists can observe what goes wrong, helping them figure out what that gene normally does. Building on that foundation, let us look at the first major category, physical mutagens. When exposing cells to a physical mutagen, like X-rays, the radiation blasts the DNA with high energy. This can break the DNA strands or fuse adjacent bases together. Interestingly, the number of mutations produced is often directly proportional to the dose of radiation received. This linear relationship is incredibly useful. It helps scientists predict genetic damage based on exposure levels, which is vital for determining safe exposure limits for radiation workers and medical professionals. This linear relationship brings us to a very important mathematical model. The formula is F equals C times D. Conceptually, this states that the frequency of mutations is exactly equal to the mutagen's strength multiplied by the total dose given. Let us break down the symbols. The letter F represents the induced mutation frequency, which is the fraction of cells that develop a mutation. The letter C is the specific mutagenic coefficient, a constant telling us how effective a particular mutagen is per unit of dose. Finally, D represents the total dose of the mutagen applied, usually measured in grays or rads. If the dose D increases, the mutation frequency F increases proportionally, meaning more radiation leads directly to more genetic damage. Now that we have our formula, let us see this with a concrete example. A researcher exposes fruit flies to X-rays. We are given the mutagenic coefficient, C, as 0.002, and the total dose, D, as 50 grays. We need to find the expected mutation frequency. We use our linear dose-response formula, F equals C times D. Substituting the values, we multiply 0.002 by 50. This gives us 0.10. Physically, this tells the researcher that if they expose the flies to 50 grays of X-rays, they can expect exactly 10% of the offspring to carry a new mutation. This allows the scientists to plan their experiment properly. Having seen how physical radiation works, let us shift our focus to chemical mutagens. While physical mutagens blast the DNA with energy, chemical mutagens are molecules that sneak into the nucleus and mess with the DNA chemically. There are a few main types. First, base analogs look exactly like normal DNA letters, but they trick the DNA polymerase into inserting them during replication, leading to mispairing later. Second, alkylating agents, like EMS, physically attach bulky chemical groups to the DNA bases. This changes their shape and causes the cell to read them incorrectly during replication. Another fascinating and dangerous class of chemical mutagens are the intercalating agents. Unlike the others that alter the bases themselves, intercalating agents are flat molecules. Because of their shape, they can slide right between the rungs of the DNA ladder. Imagine shoving an extra thick piece of cardboard into a stack of papers. This stretches the DNA structure and confuses the cellular machinery. When the cell tries to copy this stretched DNA, it accidentally adds or deletes letters. This results in devastating frame shift mutations, completely altering how the genetic code is read from that point forward. To measure how dangerous or effective these chemical mutagens are, scientists calculate the mutation rate. Conceptually, this is the probability that a specific gene will mutate in a single generation or cell division. The formula is mu equals m divided by n. The Greek letter mu represents the mutation rate itself. The letter m represents the total number of mutant cells observed in the experiment. Finally, the letter n represents the total number of cells that were exposed to the mutagen and actually survived. If the number of mutants, m, increases, while the surviving population, n, stays the same, the mutation rate, mu, increases, indicating a highly effective or dangerous chemical mutagen. Let us apply this new formula to a real laboratory scenario. A microbiologist treats a culture of bacteria with the chemical mutagen EMS. She counts exactly 450 mutant colonies, so M equals 450. The total population of surviving treated cells, N, is 1 million. We are asked to find the mutation rate. We use the formula mu equals m divided by n. Substituting our values, we divide 450 by 1 million. Simplifying this into scientific notation, we get 4.5 times 10 to the power of -4 mutations per cell. Practically, this means about 4 or 5 bacteria out of every 10,000 will develop a mutation, proving EMS is highly potent. Now that we have calculated the effects, let us visualize exactly what is happening to the DNA structure. On the left side of this diagram, you see a physical mutagen in action. A jagged blue arrow representing UV light strikes the DNA double helix, causing 2 adjacent thymine bases on the same strand to fuse together, forming a pyrimidine dimer. This creates a physical kink in the DNA backbone. On the right side, we see a chemical mutagen, a flat, purple, rectangular molecule representing an intercalating agent has wedged itself horizontally between the base pairs. you can see how it stretches the DNA ladder apart, which will inevitably cause errors during replication. We have covered a lot of ground today, so let us review the most important ideas. First, mutagens are external environmental agents that drastically increase the rate at which DNA sequence errors occur, pushing it far above the natural background rate. Second, physical mutagens, like UV light and X-rays, use high energy to physically break DNA strands or fuse adjacent bases together. Finally, chemical mutagens use chemical mimicry or physical distortion to trick the cell into making replication errors. By understanding and mathematically modeling these agents, we can better protect human health and harness these tools to unlock the secrets of genetics. Great job following along!